allowing me basically to present this work here. Yeah, as Maria said, I basically just started at Heidelberg University as a junior group leader at the center, or newly founded Center for Synthetic Genomics, that is founded across the University of Heidelberg, Karlsruhe, and Mainz. And in Heidelberg, it's incorporated into the ZMBH, where we want to use these models basically to understand gene regulation and also start using them to manipulate genomes. The work that I'm going to present today is based on projects that I started in Sarah Mostafavi's group at the University of Washington in close collaboration with the people from the Immunological Genome Project. So I would like to acknowledge these people beforehand. And a lot of the results that I'm showing will be their hard work. Like the work from Anna, Alina, Zhenming, who are PhD students in the lab, also Kat and Jan, who are PhD students in Christoph's and Jason's lab. So to motivate this talk, so basically what we get from classical association frameworks, they link sequence variants to observe phenotypes, but they only provide basically these statistical associations across the genome, as you can see here in this Manhattan plot. And so these studies usually lack the ability to disentangle the correlation between variants. They miss rare variants and the effect of combinations and cannot provide any mechanistic insights from these statistics alone. And so when we map these associations onto the genome, we can learn that the majority of these associated variants are located in regions that are non-coding, or what we define actually as regulatory regions. For example, in this study here, where people computed the relationship between genetic variants in 21 autoimmune diseases, we see that only 10% are in coding regions, 60% are about in enhancer regions, 7% in promoter regions, and then another fraction of variants in sort of regions that we associate with post-transcriptional regulation. And so to identify then causal variants in between all these associated variants, what we have to do is usually first find the cell type that is affected, and then perform additional genomic experiments for the cell type that we expect to cause this to be involved. And so here on the top, you see the association of variants in a region between two genes. And below, you see tracks of 4 genomic experiments telling us about chromatin state, transcription factor binding, and 3D interactions in that regions. And so from the overlap of this of the variant, like this variant with the transcription factor binding site, at this high C interaction of the enhancer with the promoter of gene 2 over here, we can maybe conclude that this variant up here is disrupting transcription factor binding, and so disrupting enhancer promoter interactions, which likely leads to the changes in gene expression of this gene 2. And so sequence to function models, they have basically been developed to learn the relationship between sequence and genomic measurements, as I just showed you, to directly then identify variant effects in silico. And so by training across many thousands or millions of genomic positions, they learn to identify regulatory elements in often long sequences to then correctly predict differences in the measured signal for gene expression, chromatin state, or transcription factor binding. And they do this usually by using convolutions that look like transcription factor motifs or RNA binding protein motifs that then basically enable the model to make these predictions, to remember which ones are important and then to make the correct predictions. And the cool thing about them is then once these models have learned this gene regulatory code, they can then be used to test any type of sequence variant at cell type resolution. So we can manipulate any part of the sequence and then let the model predict the effect of this modification. And if we do this then across the entire sequence, we can also identify entire cis-regulatory motifs at base pair resolution that it uses to make its predictions. And so these called sequence attributions of the model give us an idea about what parts of the sequence the model thinks are important for the measured signal. And for the people who are familiar with the models, obviously you can also do this with like backpropagation methods. But like this is, I think, like a nice way to basically describe what these attribution maps are representing. So here's again like a broad summary of what we can do with these sequence attributions. So on the left, we see we can sort of deconvolve the signal that we get in our measurements. So let's assume we had a tag-seq measurements for different cell types. We can then, with the sequence model, identify the motifs that it uses to make the predictions or to make predictions of high signal in these different cell types. So here in this cell type, it predicts that one motif is active. In this cell type, it says there's two motifs active. In this cell type, there's another one active. In this one, basically, there's probably no attack signal. So we also don't see any of the motifs that are active. And then we can associate these motifs with known transcription factors using motif databases such as SysBP or Jasper, and basically find out, come up with hypotheses, basically which transcription factor is responsible for the signal that we measure in our genomic assay. The other thing we can do is, again, like this variant interpretation, where here, this is actually allele resolved data, where we have two alleles in the same cell type, and we see that this allele basically has low signal, and according to the disrupted motif, and disrupted PO1 motif from this T to C mutation here. Whereas like in the allele B, basically we see high signal because there the motif is still intact. Okay, so while these perspectives are really exciting, these models still struggle with a lot of their core functionalities. So for example, in our paper here, we investigated how well they can determine differences in gene expression across different individuals. So we used whole genome sequencing from the ROS-map data of 839 individuals, for which gene expression was measured in frontal cortex, and investigated how well the model's predictions match the measured differences. So here, this is shown for one gene, where the correlation that is measured is pretty high. Every data point is basically a different individual. These are the observed values, and these are the predicted values for the model. So when we look at the predicted variant effects for the same gene across the window of the input of the informer model, the model clearly identifies this one variant that really sticks out here, that is driving the prediction of the model near the transcription start site and is causing these differences of gene expression across different individuals. But then after performing this for 6,000 expressed genes in frontal cortex, we observed that basically like 50% of the correlations are basically negative and incorrect. And the model missed sort of the direction of the effect, also for genes where it actually predicted like a high expression in general. And while this looks kind of a little bit that the model doesn't really understand what is going on, in a co-submission with our paper, Nila Ioannidis group basically found that actually the model is doing like a better job when causal variants are actually located within 1,000 base pairs to the transcription start site, whereas it kind of loses all the information when it's further away from the transcription start site. And this also mirrors the results from Alexander Korolos's paper from Julian's lab, suggesting that really Informer knows a lot about this promoter syntax at base pair resolution, but then really struggles with more distal elements such as enhancers. Okay, so the question basically arises like, how can we improve these models to learn more about gene regulation? So here, today, I'm trying to present two concepts that we explored in Sarah's lab during my postdoc. To some here, maybe these ideas might be more or less obvious, or you might have seen similar work, but I hope I can still present some interesting ideas and new results. So first, we can include sequence variation during the training. Now represent two scenarios, one for F1 hybrid mice, with two defined alleles, and one for individual genomes from whole genome sequencing. And then second, I will present results from increasing resolution of these models, something that also has become quite popular recently. Sorry. Okay, so first we can train models on F1 hybrid mice data. Here we actually have RNA-seq, ATAC-seq, and ChIP-seq data available. Basically, what is happening? We have these inbred mice strains that have like very defined genomes with very defined differences between each other. And so when we perform any type of sequencing assay, we can usually tell from which genome the read was coming from. And so you get basically for every cell type that you measure in this condition, in like an F1 offspring of the two, you basically get two fragments for the same location, and one you can map to the B6 genome, and the other one you can map to the Cas genome. And so you get then two signals from the same cell type. And if you have any differences, you expect that they're arising basically due to the differences in the sequence because your trans environment is basically kept constant. And so if you would use like a regular sequence to function model, you would basically train this on a single sequence. You can either train it only on the B6 genome and then also test it on the Cas genome, or if you want to be really fancy, you basically train it on both genomes, send it through the model, and basically predict the counts that you get on each of the genomes. And then if you want to predict the ratio or the differences between the genomes, so allelic imbalances, what you would have to do is then indirectly compute the difference between what you get for the B6 genome and the Cas genome. Sorry. Okay. So our contribution here is actually that instead of like training only on a signal from a single genome, we developed a model that directly predicts allelic differences from both sequences as input, and so can directly contrast the allelic ratio during training, and actually makes use then from this additional loss basically to make better predictions overall. And this is what I want to show here. The blue and the orange models are the single head models that I described at the beginning, where we only have like a single sequence going in. The green and the red one are the models, deep allele models. The green one actually doesn't use an additional head for the ratio. It simply has like a loss function that accounts for the ratio. But you can see that the best model here is the one that actually uses like this extra head for ratio predictions across 6 different data sets that we had, 2 RNA-seq, 2 ATAC, and 2 ChIP-seq data sets in predicting the ratio between different alleles. At the same time, we don't see any improvement of the model for predicting the total counts for each of the individual alleles. And so then we wanted to use that. We wanted to check if our model also is good at like understanding the mechanisms that are basically leading to these allelic differences. And so we checked this for ChIP-seq from SPI1. Up here is an example where the model's attributions clearly identifies this variant interferes with the SPI binding motif. We can also see there's actually like a Yoon motif that it also identifies to be important, which is actually a known interactor with SPI1. So it also actually learns this from the data. And when we looked at this like more systematically for all the sequences where we found that the model made a good prediction for allelic ratio, we can then see where are these like main variants here with the highest effect basically located. And most of those variants are located in SPI1 motifs. Some of them are even located in Yoon motifs, and then a couple other binding sites. So showing basically the model really picks up some meaningful biology here that is leading to allelic differences. Interestingly, then when we do this analysis for the deep allele model and compare it against the standard model with only one sequence, we observe that in general, like our deep allele model more often finds basically these main variants located in motifs. Whereas at the same time, the standard model kind of completely misses even this like very simple motif here of probably something like SPI1 or ETS or something like that, that it usually learns, but it doesn't learn to use this model here for this sequence to get any signal. And so in those cases, there's a lot of cases basically where the model fails to identify these motifs, while our deep allele model has learned additional, I would say, sequence context where these motifs are active and can be disrupted by variants between the two alleles. Okay, so then for training on personal genomes, the whole thing is a bit different because we don't have only two genomes here, but we came up with like a similar strategy that is shown here. And this work was driven by Anna Spiro, a PhD student in Sarah's lab, and Xinming Tu, another PhD student in our lab. And the way we model the data here is similar to the deep allele model from before, with a dual model input where the reference genome is used as one input, and then two alleles per individual are basically also given to the model as well. Then the model predicts mean expression from the reference sequence, and the difference of the mean from the difference of the personal genome from the mean is then predicted from the difference between basically sequence embeddings between the reference and the personal genomes. And so the convolutional layers are also shared between the personal and the individual sequences. And the difference is, again, predicted from these combined embeddings, where we take first the average of the signal from the two alleles. What is important for the rest of the story is how we evaluate basically this model. Here we need to determine different test sets. One is a test set of seen genes. So when we train across different individuals on a set of genes, we have a set of genes based on the set of genes that the model has seen, but it's in a set of individuals that the model has never seen before. So this is kind of the classical association setting, as you can see with the predict scan. And then there's a really hard case where the model has to make predictions across individuals that it has never seen on a set of genes that it has never seen. And so, yeah, when we first check basically on what happens if what happens to the predictions across different individuals for seen genes, as expected or as shown before, basically Informer and a model that was trained on the reference, so that's why that's called R, is basically performing sort of randomly as like negative correlation and positive correlation. Whereas like the model that was trained on the personal genomes consistently is able to have positive correlation across individuals that it has never seen. When we looked at this, like how big basically it does have the training set to be that the model can learn this, it kind of saturates here around like 400 people from where the performance does not improve anymore. But then, however, when we now look at the hard task of the model, looking at genes that it has never seen, we observe that there is no improvement in the predictions. So while we are training on more data, we cannot use actually this additional data to generalize here to unseen sequence variants. And then when we look a little bit more into like the training dynamics of this model, and we look at different losses here. So we're looking here at the green, we're looking at correlation across different individuals for the seen genes. The purple is like how well can it differentiate between expressed and non-expressed genes in individual cell types. And then the purple one is this hard task of like making new predictions across unknown individuals, across unknown genes. So when we look at this, so this model was initialized actually with a model that already knows, that was trained on the reference genome, on mean expression values. And the interesting thing is like after a single epoch, basically, like the model already learns for the seen genes what kind of variants are important to make its predictions. Whereas it actually then already starts to lose its ability to make predictions across different genes, kind of losing its knowledge over time of like which genes are actually expressed and which ones aren't. the same time, it never really picks up the signal for unseen genes of which kind of variants are important there to make any predictions. And so this really, I think, suggests that what happens here is that the model overfits on these variants that it can see in this set of training genes. And basically learns these seen variants here by heart, just focusing on variants with high QTLs, to use it then to make predictions for unseen individuals. However, never being able to learn something general about gene regulatory grammar here. Okay, so in summary, these results are quite different from the results that we saw from the allele resolved data from mice, where the contrastive approach really showed improved generalization. And so this might be due to the different nature of the variants that we see in the two data sets, suggesting maybe that the variants in personalized genomes are not caused by some simple transcription factor motif grammar. But we can discuss this maybe after the talk. So lastly, I would like actually to switch a bit and would like to present you another approach to improve model training and generalization, and hereby training models at base pair resolution. And so modeling at base pair resolution is actually not a really new concept. Like, in fact, it has actually been successfully applied to different data sets already. For example, it started off with like BPNet for ChIP-seq, ChIP-Nexus. CageSeq was used for CageSeq now, has been used for ClipSeq, and also for AttackSeq. And so inspired basically by these results, and also by the talks from Anshu's lab, and with also the help from Jacob Schreiber and Anusri from Anshu's lab, we felt like this was really something that we should try for our data set that we had. So before I joined Sarah's lab, Sasha developed AITech, which was a multitask CNN model for AttackSeq data that predicts the number of TN5 insertions or counts in peaks. Also, we refer to them also as chromatin regions across 81 different cell types that you can see here. And so by predicting this data matrix here, it does already a really good job in detecting known transcription factor motifs, and we can use it really to detect new biology, which they did in their original publication. But yeah, again, inspired by the base pair models that I showed you earlier, especially the chrombpnet, of course, we wanted to see if a base pair model for this data would help us with understanding the biology of immune cells better. So in AttackSeq, the TN5 enzyme cuts accessible DNA, and while it can access open chromatin regions that are open because transcription factors are binding in it, it cannot bind in the binding sites themselves as often as in the surrounding DNA. And so AttackSeq profiles contain information about transcription factor binding location, as well as the transcription factor binding strength in these so-called footprints that you can see here. Yeah, again, like the chrombpnet paper already beautifully demonstrates how we can perform deep footprinting, as I would call it, with a model like this. And I highly recommend, basically reading this paper. Here we wanted to investigate if this kind of high resolution modeling can also improve the model's ability to understand differences between highly related cell types. So Alina, who was a very amazing undergrad student in our lab, was up to the challenge, and she created now this multitask neural network at base pair resolution, which we call BPAI-Tech. We modeled BPAI-Tech after chrombpnet using a couple of convolutions, dilated convolutional layers, and then using these base pair embeddings that we get from this in two prediction tasks. One is the accessibility head that predicts the total number of TN5 insertions in a 250 base pair region around the AttackSeq peaks. And then the profile head, which predicts the distribution of TN5 insertions at base pair resolution. Since we modeled this approach after chrombpnet, we we also included the bias the bias model before we take the softmax. This takes care of the sequence preferences of the TN5 enzyme, which can change the shape of these profile so far that you can actually hide transcription factor footprints. And so this model is basically equal to the original AI-Tech model, but it has an additional prediction head for the base pair profiles. And so different from ChromaBPNet, though, like we don't use the profiles here for evaluating the performance or the interpretability of this model or to learn any biology. We're actually more interested in how well can it now predict differences between the cell types. And so also different actually from the ChromaBPNet paper, we also we are only training a single bias model here that was trained on protein-free or naked DNA from this paper here. Also highly recommend read this paper to see what beautiful biology actually one can do or discover with like ATAC-seq when you're using these ATAC-seq footprints. Whether this bias is a great choice for deep footprinting, that was already assessed and evaluated in detail in ChromaBPNet. You can see here that the naked DNA bias is not optimal for this sort of analysis since it does not model the specific bias perfectly. Again, yeah, if you want to read more about this, really check out this cool paper. Yeah, again, what we want to do is we're not interested in getting the footprints, but we're interested in looking at these two measures here. How well can it predict differences across cell types, and how well can the model predict differences within a cell type? And so for the first thing, basically, we just compared our base pair model with like optimized loss function, optimized hyperparameters, and so on against the classical model that didn't have the profile as an additional data set or data modality to train on. And I think you can clearly see that now we're actually doing way better in predicting differences across cell types. At the same time, we're also doing way better at predicting differences between regions within all of these 90 cell types that we had in our data set. Clearly showing that the model it's not some sort of like scaling issue here, but is actually like really picking up more information from the data now. BPNet already introduced this very clever approximation of the Poisson loss as a composite loss of mean squared error across total accessibility and the multinomial log likelihood for the profiles. So we also tested different loss functions here and wanted to see how they influence our prediction tasks. But what we found is basically this original loss of the multinomial log likelihood, negative log likelihood, actually doesn't perform as well as a combination of the mean squared error and cross entropies for the profiles. It's not really clear to us why this is happening. What we also see is actually if we use mean squared error or just the Poisson loss at base pair resolution counts, which we get from multiplying the profile with the total number of counts in that region, also really hinders the model to learn more about the data. So like this loss is really essential for us to get these improved predictions across cell types and across chromatin regions. An important difference also between our model and the ChromaBPNet model is that our model is a multitask model. So the hope of a multitask model is that combining related tasks helps the model with each individual task or to solve each individual task. And so to test the hypothesis, we did not want to train on 81 single models. Instead, we performed this task actually at cell lineage level. So these are the different colors up here. So these are 10, the 81, no, the 90 cell types can be distributed into 10 different cell lineages. So we basically just aggregated the data for each lineage. And then we trained a multitask model on the 10 lineages, as well as 10 individual models on each of them. And then we compared the results on the lineage level, also with a model that was trained at cell type resolution, where we then afterwards sort of aggregated the predictions from the cell types that were in the lineage. The important thing to take away here is that also here we see that both multitask models, either trained on lineages or on individual cell types, are performing way better than the model that was trained on individual cell lineages, showing really nicely the disadvantage that you get from multitasking across different related cell types here. Then we wanted to test if base pair resolution was actually necessary, because I don't know, storing data at base pair resolution takes up more space. It's a bit harder to transfer between groups, so one also needs to be processed just before training the model, basically. And so we explored the possibility to process this data in bins, because theoretically, at least smaller bins should still capture some of the transcription factor footprints, which usually bind between something like 4 to 15 base pairs. But as you can see here, basically performance of the models immediately dropped when we binned it to 5 base pair resolution, showing that you really need this base pair resolution for the model to really pick up this additional information from your data. Finally, we wanted to see if we could understand why these base pair models, these models are working better than the peak models. So we used a very stringent criteria to determine here this yellow set of sequences for which the base pair model consistently performed better across multiple initializations of the model, so that this was not just a result of some random initialization or some randomness in the model. And then across cell lineages, we determined motifs in both models, such as, for example, this one here, where the model basically, where the base pair model identifies this nicely, this RAL motif in this cell lineage, where it misses it completely, or the non-cell, sorry, the AI tech model, the classical model misses it completely. And so we then assessed the effect of the, we then extracted these motifs, clustered them. We found a couple of motifs, such as the RAL motif or the PTF1A motif, that were highly enriched in our base pair resolution model, but wasn't picked up at all by the AI tech model. And then we inserted these motifs into random sequences and wanted to see what are the differences in the predictions across different cell lineages. And we then saw what we saw already in our attributions for the individual sequence, that some of these models have like significantly higher effects in different cell lineages than they have compared to the AI tech model in blue. Lastly, I will just quickly touch on this controversial topic of the bias model. Again, yeah, BPNet did already a fantastic job to explore this in detail on the model's ability to create footprints. However, if this bias is also necessary for better predictions of the model is I think still unclear. So on top of the bias that I showed you before that was trained on naked DNA, here we also trained a bias from aggregated counts of closed regions. So this is basically the peaks that we have in our data sets, but in cell types in which they are statistically closed. And so we aggregate the signal to an aggregate signal of what would be like a closed what the signal would look like in a closed region. And then we train on these aggregated signals our second bias model. Just for to check this, what we saw is basically these two bias models from the naked DNA and from closed regions. They're really highly identical, correlating here around something like 0.85, and also on different held out data sets. These models performed really well, or like really similar. Usually the model that was trained on the same data type here, on closed regions also performs a little bit better on the closed regions held out set. But then the interesting, or like the test that we wanted to see, or what we wanted to see is like how well, how much influence does the bias model have on predicting differences across cell types or differences within a cell type. And here we saw that actually all these different bias models had pretty much the same effect. Like we still saw pretty much the same performance across cell types, but also across different chromatin regions. Interestingly, actually, is that a model that wasn't using a bias actually performed as well as the other models with the bias. And this was a bit contrary to my own intuition because I thought a model that was neglecting the bias would make the model focus more on the TN5 distribution instead of transcription factor footprints. But it seems like it has enough capacity to basically pick up both. And since all these models seem to learn the same information from the profiles, then we were wondering if also the information that we can extract from these models is then also different. It's also similar. And then, so we looked again at a couple of attributions across cell lineages, and indeed we found that when we looked at attributions of sequences with similar predictive performance in both models, we saw that the motifs from the model with the bias and the motifs or the attributions from the models without the bias were actually really, really similar. So I would conclude that if you are not interested basically in creating these deep footprints from your model, you probably don't need to work with a bias model in this case. What is more important is that you work with this dual head architecture that is able to disentangle the profile contributions from the accessibility predictions, because otherwise you might then get the TN5 bias into your attributions. And all of this is also already shown in the ChromeBPNet paper. Okay. Yeah, in summary, I tried to show you today two advanced strategies to train sequence-to-function models. First one is I presented a method to train these models on sequences with variants. So while this approach looks promising for F1 mice, we saw that it overfitted for personal genomes. This might be due to the differences in the kind of variants that we observe in these two data sets. Then I hope that I was able to convince you that base pair resolution training not only returns very nice footprints, but also increases the information that your model can extract from your available data. We also showed that multitask learning improves knowledge of the model and confirmed that ChromeBPNet's observation that attributions from the total accessibility are actually not affected by the TN5 bias. And yeah, with that, I would like to thank Sarah again and everyone in the lab who was involved in this, especially Anna, Jinming, and Alina, who were the main drivers of these projects. And I would also like to thank the people from ImGen who have created and curated these amazing data sets for us over the years. They also helped us with data processing and the analysis. And lastly, yeah, I would like to advertise a little bit for myself. We still have an open position in my group. We want to understand the mechanisms that control translation, sort of the next layer of gene regulation. So please, if you are currently looking for a place, please send us an email. Thank you very much.